Hi, everybody. Chapter 15. These are our senses, and we're going to focus on one, our sense of vision. If you've noticed that I've kind of lost a little bit of my lecture fun lately, so I get that I'm like, oh, they're so tired of listening to me talk. And when I start thinking that, then I don't know, starts bumbling. So hopefully I can get it together in chapter 15 and be super excited and help you learn this material about senses. Okay. All right. So don't need an overview of the nervous system again, but we do know that we have the sensory division of the peripheral nervous system, and it is making up our senses. So we think about senses, it's our sense of touch, pain, taste, smell, vision, hearing, all the things, right? So we have two kinds of receptors. Now, a receptor is usually free dendrites. Okay, so if you go back to your neuron, it's free dendrites that are acting as little sensors, if you will, detecting the stimuli in our environment. Now, this stimuli can be coming from outside the body, and we call that exteroceptors. And so outside your body, so that could be the light from my lamp, or the colors that I'm seeing, or the taste of my Diet Coke, or if I'm touching something, my sense of touch. Interoceptors are inside. So a really big one is our pressoreceptors, also known as baroreceptors, essentially detecting pressure. So pressure in our arteries, pressure in our bladder. Pressure is a big one. So when we really think about that, when we have fluid under pressure, like in our cardiovascular system. Osmoreceptors for water and salt balance, chemoreceptors monitor the pH of the blood, and you can see essentially that internal receptors or interoceptors are super important to maintaining homeostasis. So exteroceptors are sensing outside stimuli, interoceptors inside. So not so hard. Here are some categories of sensory receptors on the far left-hand side of this table, and what kind of stimulus they are responding to, and the category of exteroceptor they belong to. Our taste cells and our sense of smell, so that's olfactory, are both what we call chemoceptors or chemoreceptors, and they're associated with the taste bud and the olfactory lining, respectively. We have rods and cones on the back of our eye and the retina, detecting light, and these are a type of photoreceptor, photo meaning light. Of course, it's sensing vision associated with the eyeball. Hair cells, which we're not going to get into that, but there are hair cells in the inner ear that are responding to sound waves, and that allows us to have our sense of hearing. It's called a mechanoreceptor. We have some hair cells in our semicircular canals to tell us where we are in space. If we're sitting up, lying down, or spinning, like in an amusement park ride, another type of mechanoreceptor. And then lastly, hair cells determining where we are in space. So here are those things outlined a bit. So chemoreceptors respond to chemicals, taste, smell, and blood. Nociceptors or nociceptors, these are our pain receptors. We talked about photo, mechano, and thermoreceptors. These are super important to tell you essentially like what is the temperature. If you're about to maybe take a shower and you put your hand under the water to see how hot the water is, we have hot and cold receptors, a type of thermoreceptor in our skin that's going to help us detect the temperature of that water. If we walk outside and it's too cold, we're going to determine that temperature by these thermoreceptors, and so we put on a jacket or hat or whatever. Okay, so sensation is a weird thing. So I know in the previous chapter we talked about this whole idea of having an effector. So when we go the whole way, so we go from the sensory nervous system to the central nervous system to the motor nervous system, having some effect. Well, when we have sensation, essentially what's happening is you go from your receptors to essentially the spinal cord. So we go from a sensory neuron to an interneuron to the brain, and we essentially have the sensation because of the activity in the cerebral cortex. You don't necessarily have that information going back down the spinal cord to the motor division or the motor part of the peripheral nervous system. So essentially, sensation is fairly quick because you don't have to go back out to the motor division. Okay, so pretty simplified diagram. Here we have the stimulus, these free dendrites, whether that's the thermoreceptors or baroreceptors or photoreceptors. And this is going to be detected by a sensory receptor. So you already know that. Sensory receptors are part of the sensory division of the peripheral nervous system. And then that's going to travel along nerve bundles, essentially, to the spinal cord. So now we're in the interneuron land of things. And those bundles of interneurons forming this massive nerve, your spinal cord, takes that to the brain. We don't go back out again because it's a sensory perception. So in the cerebral cortex, depends on where it is, we're going to have that sensation. So here are some particular sensory receptors of the skin. So your book calls them cutaneous receptors. I just thought it was interesting. You don't have to know all these names except for, hey, look, free nerve endings, pain, heat, and cold. So there are hot and cold receptors. We have a particular kind of dendrites that are playing a role in touch. So we have touch here, the Krausen bulbs and the Merkel discs, touch, root hairs, touch, the Meissner corpuscles, and so on, touch. Then we have 2 for pressure. So there's a lot of different kinds of receptors in the skin. And I just tell you that because that's why the skin is so sensitive. You don't need to know all those different examples. It's just pretty cool. So the one sense that we want to dive into and the organ that's involved is the human eye. So let's take a look at the eye. Again, a picture is worth 1,000 words. And I want to start with the optic nerve. Okay, so every one of our sense organs has a specialized nerve that goes to it, and it's coming directly from the cranium. So they are cranial nerves. So the optic nerve is one of the cranial nerves. There it is. And associated with the optic nerve is essentially the back of the eye. Now, there's something really interesting going on with the back of the eye because along the back of the eye, we refer to this as the retina. But there is an area here we are going to call the blind spot because where the optic nerve exits the eye, there are no rods and cones. There's no way to see there. So that is called our anatomical blind spot. So let's go to the front of the eye. So we all have a covering over our eye called the cornea. And inside the cornea, then we essentially have a compartment filled with what we call the aqueous humor. It is a watery type liquid that is here, and it is going to help with refraction. So as the light is coming through our pupil, then that aqueous humor, along with some other parts, are going to make sure that the image then is going through the pupil and through the lens. Now notice that the lens is associated with these ligaments, suspensory ligaments, which are going to help change the shape of the lens by being directed by the ciliary body. So the ciliary body is going to essentially either pull those suspensory ligaments or allow them to relax. And so this is going to change the shape of our lens. So it's either going to be really rounded or less rounded. This is important because as that image comes through your pupil, the eye, this part of the eye, lens, suspensory ligaments, and ciliary body are essentially working to make sure that that image lands on the retina so you can see it. So the retina, this inner layer of part of the back of the eye, then we have a choroid and the sclera. So this is a fibrous layer on the outside of the eye. And the choroid, I believe, is filled with some rods and cones. Maybe we'll come back to that. Okay. All right. Sclera. So that's that hard, white, fibrous part of the eye, protecting and supporting the eye. The cornea, of course, that's the covering of the eye, refracts the light rays. The pupil allows the light and the image to go through. The choroid, I think I said something about rods and cones. It's actually absorbing any stray light that is not directly being sensed, if you will, by the retina. The ciliary body is going to hold the lens in place. Of course, we know there's some ligaments that are going to help it move. The iris then is this color pigment around your eye. And the iris, it like shrinks or expands based on the light outside, and it's really causing the pupil, which is just dead space, to either become smaller in bright light or become larger in less light. So the iris, the pigmented part, is actually controlling the pupil. The back of the retina, we have our photoreceptors, so those are our light receptors. We have the rods that are going to detect black and white and really are responding, I said black and white, but really are responding to brightness. And they work really well in dark. So when you're driving at night, your rods are pretty active. Cones are for color. So essentially we have three kinds of cones: red, green, and blue, just like you have RGB on your computers. So we have three colors that are going to mix together to make all the variety of colors that we see. We're going to skip that one. The lens, of course, refracting and focusing the light rays. We'll come back to that. The aqueous and vitreous humor. So the aqueous humor is in the front of the eye behind the cornea. The vitreous humor is like filling in the back of that eye behind the lens, and they're both essentially fluids that are helping support the eye. And then, of course, we have the optic nerve. Okay, so visual accommodation. Have you ever had someone, take a piece of paper and just go, "Hey, look at this"? Like kids are really good at that. "Hey, mom, look at this." And you're like, "Whoa, I can't even see that." You can see it probably eventually, unless you're, over the age of 40, in which, you have trouble with what we call accommodation. Accommodation is when you're able to look up close and look far away and look up close and look far away. And you know what? Your lens changes shape pretty quickly so that you can focus. And that image is landing directly on the retina. So as I mentioned earlier, I didn't say there was a ciliary muscle, but essentially the ciliary muscle, which is within the ciliary body, is changing the shape of that lens. So if you're looking at a distant object, the ciliary muscle is relaxed and the lens is flat so that the image lands right on the back of the eye. But if someone takes a sticker and says, "Hey, look at that," and it's right up, up on you, then essentially what needs to happen is we have to contract those ciliary muscles, and we need to round out that lens so we can get the image to focus at the appropriate spot. So when we start having problems with visual accommodation, then guess what? We have to use some glasses to help us. Yeah. Okay. All right. Let's take a look. I've been describing this, but in figure A, focusing on a distant object, then we can essentially flatten this lens, and the image is coming through and it can hit the retina. But when something's up close, then we need to essentially contract those muscles of the ciliary body and cause that lens to round. So now we can have that near image landing on the appropriate spot of our retina. So here's some rods and cones under electron microscopy, and they've colored it to make it look cool. Okay, there we go, our rods and cones. Okay, talked about that. There's a blind spot. So let's talk about distance and near vision. So you're going to learn more about this in your lab. But essentially, if you are nearsighted, so you can see close but not see far away, your eye is elongated, so your distant objects are brought into focus in front of the retina. Let's take a look. So here you are, nearsightedness, and so your image is landing before the retina. So you use corrective lenses to essentially bring that image to the retina. So we use a concave type lens. If you're farsighted, so you can't see close up, the eye is too short. So if the eye's too short, then we need a convex lens so that we can bring that image up closer